A point mutation in the DNA is a substitution of one nucleotide base for another. Here we have a wild-type DNA sequence that codes for a protein consisting of 4 amino acids. The coding strand of the DNA is shown on the top in the 5 to 3' direction. During transcription, RNA polymerase reads the template strand in the 3 to 5' direction and adds nucleotides in the 5 to 3' direction. The resulting sequence is the mRNA that will be used in translation to make a protein. During translation, sequences of mRNA that are 3 bases long are used as codons to either start or stop translation or to code for specific amino acids. This particular mRNA sequence codes for a protein made up of tryptophan, isoleucine, threonine, and cysteine. The final protein is this sequence here. If a point mutation were to occur in the original DNA sequence, there could be a missense, nonsense, silent, or read-through mutation. A missense mutation causes a change in the amino acid coded for by the mRNA sequence. Here in the original DNA coding strand, one of the bases is thymine. If a mutation occurs and it is replaced by adenine, it will cause a transversion. This change will cause a different amino acid to be coded for by the mRNA sequence. Instead of the AUU sequence in the mRNA coding for isoleucine, we now have AAU, which codes for a different amino acid, asparagine. This single change in the coding strand of the wild-type DNA causes one amino acid to change, which may alter the protein's function or make it non-functional. The protein now consists of tryptophan, asparagine, threonine, and cysteine. Another mutation that can occur is nonsense, which is a point mutation that results in a codon becoming a stop codon. Here in the original DNA coding strand, one of the bases is cytosine. If a mutation occurs and it is replaced by adenine, it will cause a transversion. This change will end the production of amino acids early. Instead of the UGC sequence in the mRNA coding for cysteine, we now have UGA, which is a stop codon. Since the stop codon was translated after only 3 amino acids were made, the resulting protein only consists of tryptophan, isoleucine, and threonine. The protein is now missing an amino acid, which may alter its function or cause it to be non-functional. The next mutation that can occur is a silent mutation, which causes the mRNA sequence to code for the same amino acid that it would have if there were no mutations in the original DNA. In this DNA coding strand, one of the bases is thymine. If a mutation occurs and it is replaced by cytosine, it will cause a transition. There will be no change in the amino acid because ACU and ACC sequences both code for threonine. The resulting protein is the exact same protein that was coded for by the original DNA sequence. A fourth mutation that can occur in the DNA is read-through mutation. This point mutation occurs when a stop codon is changed to a codon that codes for an amino acid. In this DNA coding strand, one of the nucleotide bases is adenine. If a mutation occurs and it is replaced by guanine, it will cause a transition. This change will keep the amino acid sequence from stopping after cysteine because the next codon will no longer be a stop codon. The mRNA sequence will have the UGG codon, which codes for tryptophan instead of the UGA stop codon. Another tryptophan will be added to the final protein because the mutation in the stop codon changed it to a codon for tryptophan.